Hello everyone. Let's start the chapter 13. So the immunity actually so the immunity actually is adaptive immunity, okay? Because so when we get infections, actually the infections will induce our immunity response uh, to activate the B and T cells, either produce more antibodies or attract T cells, phagocyte the pathogens. So this is the immunity. So the ability of the body to react with foreign substance is called immunocompetence. So how the pathogen can induce our immunity? Because pathogens contain antigen. So the antigen is the foreign molecule that can be recognized by our immune system. So since antigen can induce the immune response, so this is also called immunogen. The substance that can work as antigen are the proteins or the polysaccharides molecules. So once the pathogens invade our humans, so their foreign proteins or polysaccharides can induce our immunity response. So the immunity is very specific because one type of antibiotic only react to one type of pathogen, such as the antibody for the chickenpox cannot against the measles. Memory, because the lymphocyte can memorize the the immunity response. So when the pathogen invade our human again, so the lymphocyte can recognize the pathogen very quickly and initiate the response very quickly. So the immunological development includes the lymphocyte development and local deletion, presentation of antigen, challenge B and T cells response, and B cells response. So all lymphocytes are derived from the hematopoietic stem cells. So B cells matured in the bone marrow side, T cells matured in the thymus. So both cells migrated to the lymphoid organs. So T cells, B cells are circulated in the bloodstream, and so they can also migrate to tissues to to perform the immunity response. So this diagram shows maturation of the T cells and B cells. So the T cells have to contact with antigen presentation cells, so they can get the immunity information and derive it to the different T cells. So the B cells actually can also need to get the immunity information from the T helper cells and then divided to plasma cells or the memory B cells. So how the antigen information is presented to the lymphocytes? So actually, this task is performed by the antigen presentation cells. So those cells are the cells with phagocytosis capacity. For example, so the recent phagocyte can digest the pathogens. So those cells would be the macrophage. And even the B cells can also work as the antigen presentation cells. Once the macrophage digests the pathogens, so they can degrade the pathogens, get the small piece of antigen, and then so they can deliver the antigen to the lymphocytes. So we have three type of antigen presentation cells. So all the cells actually have different markers. So these markers can be used for the detection, recognition, and cell communication. So these markers actually are very important for the immunity. This marker. Let's talk about the first important markers, such as major histocompatibility complex (MHC). So this is a cluster of genes that can code these markers. So these markers are the series of glycoproteins found on all the cells except red blood cells. So they are also known as the human leukocyte antigen. So these markers are very important for our immunity to recognize the foreign substance. So it also involved in the rejection of foreign tissue. Okay. So it has three types. Class 1. Class 1 can be found in all the nucleated cells. So it is used for recognize ourselves. Okay. Class 2 can be found on the macrophage downstream cells, B cells. So they are found in the antigen presentation cells. Class 3. So it's class 3 is involved in the complement system. So this is class 1 and class 2 MHC. Now, second is the CD molecule. CD means the cluster of differentiation. Okay, so we have more than 300 CD molecules. So they are involved in the immune response. So the most important are CD3, CD4, CD8. Lymphocyte receptors. So the function of lymphocyte receptor is to accept and grasp the antigen from. For example, the B cells have the receptors that can bind antigens. Okay, T cells also have the receptors that can bind antigens. So once the receptor bind to antigen, so it can induce the immune response. So B cells, T cells, they can initiate. So they can initiate the immune response. However, their responses are different. So how T cells respond to antigen? So T cells have different types. For example, we have the helper T cells, regulatory T cells, and cytotoxic T cells. Helper T cells can activate macrophage B cells process and also can activate cytotoxic T cells. Regulatory T cells can control T cells respond to the anti-inflammatory cytokines. Cytotoxic T cells will kill the infected host cells and other foreign cells. So regulatory T cells can secrete cytokines. Okay, so these cytokines can help kill the pathogens. Our B cells, after activation, B cells will become to the plasma cells. The plasma cells can produce many antibodies to react on the foreign antibodies, foreign substance. So antibodies can attack the antigen, can bind to antigen and attack the foreign substance. Now let's work on this question. B cells mature in and T cells mature in both cell types that migrate to an. So I give you 5 seconds. The question answer is D. Okay, D. Now, so this is the table shows the difference between the B cells and T cells. We can see the B cells mature in the bone marrow and T cells mature in the thymus. T cells produce antibody immunoglobulin, and T cells normally don't secrete the antibodies. So T cells is widely found in the bloodstream. Okay. So B cells use antibodies to kill the foreign substance. However, T cells can use their cytokines or directly kill, engulf the foreign substance. So we have talked about the CD cluster. Okay, so CD3 can be found on the T cells. CD4 is also found in the T cells. So this is found in the helper T cells. CD8 actually is found in cytotoxic T cells. So we can see this is surface of T cells. Okay, so we can see here. So this is CD4. Okay, CD4. So this is antigen binding site. Okay, and so CD3 actually can be. So this is MHC1. Now the B cells development. Actually, when the lymphocyte is generated, so they are the naive lymphocyte. Naive lymphocyte doesn't have any functions. Okay, naive lymphocyte will secrete in the blood, and then so they will home to specific site. Okay. So where they can attach to specific molecule and become and can differentiate to the plasma cells. So this is surface of B cells. So once T cells, B cells reach to the lymphoid tissues, so they have to so they will exchange the antigen information with the antigen presentation cells. Okay, and then they can differentiate to the matured cells. So they will so the T cells, the B cells will produce antigen receptors on T cell, B cells. So the antigen receptors actually can be assorted in different ways. So they can have different they have many different receptors. So this is a mechanism of antibody variation. Okay, so because the gene code for antibodies can be assorted in different ways, so can be generated even from same stream DNA. However, so the assortment of the RNA transcript can generate different animal side sequence. Okay, so cause a huge diversity of the antibodies. For example, let's talk about the BCS receptor. So this is immunoglobulin. So this is actually this is a large glycoprotein, and large glycoprotein serve as antigen receptors. So the immunoglobulin contains antigen binding site and variable region. Antigen binding site, so it's like a fork-shaped proteins. So the binding site actually has huge diversity shape. Okay, so that can allow the immunoglobulin can bind all kinds of antigens. So the variable region, so this area extremely versatility, okay, from one clone to another clone. Immunoglobulin also have the light chain, heavy chain. Okay, so we talk about it. TCS receptors. So it's also one type of immunoglobulin. Okay, similar to the receptors of the BCS. So it has all a huge diversities. Okay, and it also has agent, so the agent variant region and the constant region. So the clonal selection is involved in the TCS BCL differentiation. Okay, because once the TCS BCS are produced, so they have to be selected for mature. Okay, so if the our body uses the clonal selection to select the good TCS BCS for the maturation. So the lymphocyte specificity actually is pre-programmed. Okay, pre-programmed. So each lymphocyte actually express only a single specificity. However, the clonal so the TCS and BCS. So once the TCS BCS contact the antigens, so they can be activated to many different clonal expansion and many different clonal type. Okay, so this is called a clonal expansion. However, so our body have to identify the TCS and BCS because if TCS BCS some TCS BCS can recognize our our body CS antigens. That means those TCS BCS can recognize our body CS as foreign CS. Okay, so once those CS matured, so they will attack our body host cells. So that's not good. So our body uses clonal direction to eliminate those T cells, B cells, to eliminate those clones. Here we can see, for example, so this is T cells, okay? So once the T cells, the lymphocyte cells, when they matured, so they accept all kinds of antigen and receptors, okay? However, if the receptors can recognize our host cell, so that means once this lymphocyte matured, so they will attack our host cell. So that's not good. So those lymphocytes will be eliminated, okay? So if not, we will have the autoimmune diseases. So now let's work on this question. For each of the properties listed, determine if it describes T cells, B cells. So mature in the bone marrow. So this is B cells, okay? So high number in the blood, so this is T cells. Require antigen to present with MH molecule. This is T cells. Produce antibody, this is B cells. Selected help the other immune cells suppress killed, so this is T cells, this is help T cells. Now let's talk about the antigen presentation cells. So how T cells, B cells get the antigen? Because we have several specific cells that can deliver the antigen. So antigen should be so the antigen could be the whole microbe, okay, or part of the microbe, or even the cell substance from our humans, okay. For example, the blood type, okay, ABO blood type can, so they are the antigen host, human host cell antigen. Complex proteins or proteins containing molecules. So the antigen could be the protein or polypeptides, lipoprotein, glycoprotein, nuclear protein, such as DNA complex to the protein, okay, polysaccharides and lipopolysaccharides. So good antigen should be the antigen can induce immune response, and the good antigen. So they have their specific chemical composition to induce specific reaction. So they have their ideal size to induce reactions. Okay. So they can also induce a specific immunity response to generate specific cytokines or antibodies. So large antigen are better than small antigen because larger size antigen easier to induce immune response. So only large size is not enough. So if the structure is complex, so that's way better than the large size. For example, even we for the polypeptides or the polysaccharides, even so they are large size. However, they have sample structures, so they are not considered as antigens. Okay, so even the pollen is small. However, the pollen has the complex structure, so it can be a good antigen. Heptin are the small foreign molecules, so they cannot induce immune response. because they are incomplete molecules for the immune response. However, when the heptans bind with other molecules, so those combination can induce immuno response. So actually the function of the heptan can increase the epitope of the antigens. For example, the drugs, metals, or other substance can work as the heptans. So once they bind with serum proteins, so they can work as antigen to induce immune response. Alloantigen, that means the antigen from our host cells, okay, like the ABO blood type. Super antigen, normally they are the bacterial toxins. So they can induce huge immune response, for example, to activate the T cells. So they can induce the huge releasing of the cytokines and steroids that can cause toxic shock syndrome, okay, and some autoimmune diseases. Allergens, that's antigens that can induce allergic reactions. Now let's talk about the antigen presenting cells, okay? And this is also referred as the APC. So we have three types of APC: macrophage, B cells, dendritic cells. So those cells actually can digest the pathogens and degrade the pathogens to make a small piece of antigen, and then deliver those small piece of antigen to the T cells. Okay, to the T cells. Yeah, here we can see the macrophage can engulf the pathogens and then digest the pathogens to make the small piece of antigen, and then deliver this antigen to the T cells. So most antigen will be presented to the T cells. And then, so the T cells can use this antigen information to for the differentiation or either activate the other cells or the B cells. So we have some antigens. So those antigens actually doesn't need present to the T cells. Okay. So those antigen can directly activate the B cells. Okay. So these are the normally they are the carbohydrate substance like polysaccharides from E. coli and the polysaccharide from the Streptococcus pneumonia. Some molecules from the rabies and EB virus also can work as this type of antigen. Now let's work on this question. Which of following is not an antigen presenting cell? So we can see, so macrophage is, right? Dendritic cells is, B cell is, so T cells, T helper cells is not, okay? So the correct answer is C, okay, is C. So T cells response, okay, T cells, so T cells response is complex. So T cells actually is activated by the APC when they receive the antigen information. However, they also requires MHC recognizing and to eliminate the potential CF recognition. Okay, and then the T cells can produce the cytokines to attack the pathogens or induce other cells maturation. Okay. So we have different types of T cells. T helper cells, so it's CD4 positive, okay. T helper 2, CD4 positive. T helper 17 is also CD4 positive. T follicle helper cells is CD4, CD4TL. T regulatory is CD25 and 4. So these are the all kinds of functions of these CD cells. So T helper cells, so all these T helpers are CD4 positive. So they are critical in the regulating immune response. Okay. So they can activate the macrophage, okay, or active macrophages through the receptor binding, or either active macrophages through the interferon gamma, okay. T capsule can secrete the interleukin 2. Interleukin 2 can stimulate activate the other T cells. Also, it can secrete interleukin 4, 5, 6. So those can stimulate the B cells. So T helper cells is the most abundant. Okay, so it is 65%. So here we can see the T cells activation, and then so it's cytotoxic T cells. Cytotoxic T cells actually can kill the infected host cells or the cancer cells. So they can attach to these cells and kill those cells. Gamma T cells. Gamma T cells also have the T cell receptors. It can recognize a bunch of antigens. So gamma cells can respond so quickly and have the memory. Okay. So it can also attack the tumor cells. So it's considered as the bridge between the non-specific and the specific immune response. So let's talk about the other cells, natural killer cells, NK cells. So NK cells is not a specific immunity response. So these cells circulate in the blood, so they can kill the cancer cells or all kinds of infected cells. Natural killer T cells. So it's like hybrid cells. So those cells have the characteristics of both NK cells and T cells. So it is also considered as the bridge between the non-specific and the specific immunity. Now let's work on this question. Cytotoxic T cells are programmed to kill all following expect. So I'll give you 5 seconds. So cancer cells, so it can kill, okay, virus infect, foreign graft, yes. So bacteria, okay. Now let's talk about the BCS activation. So this is BCS activation. So the B cells actually, eventually B cells will become to the plasma cells and produce antibodies. So the antibodies have the antigen binding fragment, okay? And then antibody have the crystallizable fragment. So that's a pair of identical heavy chains bound together with disulfide bonds. Also pair of identical light chains. So there's one light chain bound to one heavy chain with disulfide bonds. So the linked region between the antigen binding fragment and crystallizable fragment actually can smear the and let the antibody bind twist and binding. Okay, so this is the diagram of the antibody. Okay, so we can see this is a heavy chain. This is the light chain. Okay, light chain. So this is variable region. Okay, so they can bind with all kinds of antigens. So this is constant region. So this part we are binding to the cells. We have carbohydrate elements here. They can bind to the plasma membrane. So the antibody has a hypervariable region, okay? So it means the shape of the region actually is very diverse. So it can allow this region to bind all kinds of antigens. Because the animal acids remnants in this region is very variable, okay? However, since the shape, so there are all kinds of shape actually. One shape is specific for one type of antigen. So this is the specificity of the antibody. So the specificity on two FAB set is identical. Okay, it's identical. Since the antibody can bind with the antigen, so the function would be so they can bind to the microorganism like bacteria to inhibit the bacteria. Okay, and then so they can induce, so they can guide the bacteria to the engulfment of the immune cells. Okay, so they can also bind with the virus to neutralize the virus and let our cells to destroy the virus. They can also link the cells to cause agglutination. For example, when we infuse the blood, okay, blood transfusion. So if we make a mistake, transfuse the wrong blood type of blood, so it can induce agglutination. Antibody can also bind with the microorganism, cause the microorganism lysis. Okay. Obviously, antibody can also bind with foreign substance like the toxins to work as the antitoxins. Now let's talk about the crystallized fragment. So those fragment actually has a constant structure. So this fragment actually can bind with all kinds of the immune cells, such as the macrophage, neutrophil, eosinophil, mast cells. So this fragment actually links the antigen and the microorganism to the phagocyte. Okay, so this is called opsonization. So since antibody actually is one type of immunoglobulin, okay. So for example, if IgE bind to the basal cells, mast cells, so they can cause those cells to release the allergenic mediator, cause allergy, okay. Now let's talk about different immunoglobulin. So first, let's talk about the IgA. So we have two forms, okay. One is this called isotype. One is monomer. Monomer is just one molecule of IgA. Dimer means two IgA linked together to form a dimer. So the dimer is widely found in our body, okay, such as in the salivary glands, intestine, nasal, breast, in the milk, okay, lung, and genitourinary tract. So the IgA can coat the surface of the duct, okay. So they also exist in the saliva, tears, milk, mucus, so they can bind with the microorganism to and kill the microorganism. Colostrum is the earlier secretion of the breast milk. So colostrum contains a high concentration of IgA. So that's a good source to provide the immunity for the infant. So this is the table shows the different immunoglobulin. So the level, so the concentration of antibody in the body fluid is called titer. Okay. So once the BCS is activated, so BCS can produce the microglobulin. Actually, once BCS contact with antigen, so in the beginning, so BCS needed sometimes to start the production. So this is called a latent period. So there's no immunoglobulin production, and then so the BCS can secrete the immunoglobulin. So BCS will secrete the IgM first, okay, and then IgM level decrease, so BCS start secrete IgG. So BCS can secrete IgG for a relatively longer time. And then, so once the infection is gone, so the BCS will stop the secretion. However, the BCS have the memory to the antigen. So once those pathogens invade again, so the BCS will immediately start the production of the immunoglobulin. So we can see they can directly produce IgG first, okay, and for longer time. Now let's work on this question. Match each antibody type with this description. So we can see IgG is for the allergy, okay. IgG is for most prevalent antibody. IgM is the first antibody production. IgA is secreted on the mucus, okay. IgD is the receptor. So the immunity, actually we have natural immunity and artificial. Natural immunity, for example, so that's immunity induced by the infections, okay, or the foreign substance. Artificial immunity, that means we directly get the antibodies from other resources. Okay, so those antibodies directly provide the immune response. So the active immunity, for example, so that's foreign substance or pathogens directly induce immune response. So active immunity actually requires several days to develop. Okay, so that means so we have the latent period. Okay, however it lasts for long time. So so active immunity can be stimulated by the natural or artificial means. Okay, natural, that means infections. Artificial means the vaccine. Okay. So passive immunity, actually, so that means we just directly get the antibodies from our human or animals. So those antibodies provide the immunity response. Okay. So we don't have the immune response. Since this passive immunity doesn't have the latent period. Also, this type of immunity doesn't have the memory. Okay, so our immune cell doesn't product the antibody. So this is short time. Okay, because once these antibodies are gone, so this immunity disappear. So, for example, the natural immunity. So if we get the infections, so we get natural immunity. So this is passive immunity. However, this is also nature. Okay, from a mother. So the artificial, that means, for example, we got the vaccine. Okay, vaccine, and we transfuse the antibodies from other human or animals. So the vaccine, so the immunization history, we have a long history for the immunization. So the first recorded attempt is the 6th century China. So the Chinese, ancient Chinese tried to grab up the smallpox crab and blow them to the natrium of the people, to let these people build up the immunity against smallpox. So the variolation is first variolation is was in the 10th century. So this is the people try to inoculate the dried pews from smallpox of one patient to the healthy person, to let the person get the immunity against the smallpox. So in 1796, scientists got the idea from the dairy maid, because dairy maid get the cowpox, so they have immunity to the smallpox. So the scientists let the healthy people to be inoculated with cowpox, and then so the people can get immunity from the cowpox against the smallpox. So this type of vaccine help people to get rid of the smallpox. Passive immunization, for example, we transfuse the horse serum that contains antitoxins to prevent the tetanus and diphtheria. Okay. So until now, we also we also might use the gamma globulin. Okay, so this extracted from the human blood. Okay, so gamma globulin actually can be used to treat the hepatitis B and HIV. Okay. Now let's talk about the vaccine. Vaccination belongs to the artificial active immunity. So it can stimulate the immunity response. So it has the memory response. Okay. So once the healthy people get the vaccine, so they have the potential immunity reactions to the further expose of the real pathogens. So the vaccine helps our human to get rid of many kinds of infectious diseases. So how the vaccine is prepared? So it is used to protect against the nature-widely formed pathogens. The vaccine actually has no side effects. So it can stimulate both B cells, T cells. So the effective period is long. Okay, lasts several months or several years. Doesn't need too many doses. Okay, cheap and no expensive. So we can use live attenuated cells of virus or killed cells virus to make the vaccine. We also can use the antigen fragment of the cells and the virus to make the vaccine. Conjugated vaccine means so we can conjugate the several pieces of antigen to make the vaccine. For example, we can see this is a whole cells vaccine. We can use the killed or attenuated cells or virus to make the vaccine. We can also use the antigen fragment to make the vaccine. So DNA vaccine means we can conjugate the antigen information DNA to the plasma and then inject the plasma to the human. So the human can translate the DNA information in the plasma to produce the antibodies used for the vaccine. So we can administer the vaccine through the subcutaneous route, intramuscular route, and intradermal route. Okay. So the nasal and oral route is really rare. Okay, it's really rare. So the Java is a substance so that actually required by the vaccine. So the Java can enhance the immunogenicity. Okay, also can prolong antigen retention at the injection site, facilitates antigen presenting cells engulfing. Okay, so most adjacent are aluminum hydroxide salt. Vaccine is very safe. However, vaccine can induce local reactions like swelling, warming, also can induce fever, allergy, or other more severe side effects. However, those side effects are mild or benign. Okay. So the serious side effects are really rare. Okay. However, some scientists found that vaccine is related to the diabetes, asthma, autism. However, those studies are not true. Okay, are not true. So some physician or scientists who discovered this unreal conclusion has been punished for their mistake. Okay. Actually, so the vaccine is safe. Actually, it's not related to the autism. So the vaccine actually is relatively inexpensive. So now vaccine is widely used. Now let's look on this question. Determine which of the statements below describes the active and passive immune. So infusion of gamma globulin is passive. Okay, recover from influenza is active. Recipient of the influenza vaccination actually is active. Okay, antibody produced from mother to the infant actually is passive. Okay, so this is end.